uh, understanding and mapping out the regulatory circuit in the immune system. She's majorly passionate about medicine, biomedical and clinical research. And Kumba is also interested in pursuing research on the intersection between neurology and infectious disease. She's hardworking, solution-driven, and she majorly volunteers with organizations to help in fundraising for health and humanitarian relief. And she also serves as a mentor to kids. So today we are really glad to have her join us today for today's session. And today she'll be speaking on understanding male and female immune differences to viral infections and vaccinations. So once again, I want to say welcome, Kumba, you're welcome to Science Cafe, and we're really elated to have you join us tonight to share with us. So I'm going to be handing over to you now to begin your presentation. Thank you.
Thank you very much.
Yeah, thank you. You're welcome.
For the introduction, and I am going to share my screen and make sure that everything is working well. So is my screen appearing okay?
Yes, it is. Yes.
Okay, great. So hello everyone, and thank you for being here. Thank you for your time. And I also want to extend major thanks to Science Cafe organizers. I've mainly been talking to Martin, but I know there are several other people too. So I'm really, really honored and delighted to be here today to talk to you. And I hope that this next 1.5 hours will be really great, engaging in sharing who I am, my science, and also like some of the things that drive my interest, my scientific interest, and then also my community work. And if at any time during the presentation you have a question, please feel free to say it. You can put it in the chat, or you can unmute and ask, whichever way is possible. So I would appreciate that. So yeah, I am Kumba, so I'm just going to give a brief introduction. So that will set the ground, as I said, for my interest. So originally, I am from Freetown, Sierra Leone, which most people in this call may be familiar with. And I grew up in Sierra Leone. I was born there. I went to school there. Particularly, I went to the Annie Walsh Memorial School, which is the oldest all-girls school in sub-Saharan Africa. So I did both my WAEC there, so my basic education certificate examination, which is GSS 3. And then also I did my WAS, so the West African Senior School Certificate examination there. And while at the Annie Walsh, I was the head girl, and I was very driven in wanting to do science, wanting to use science to also, I would say, address some of the questions that were in my community, as most of us may be familiar with. The burden, especially of infectious diseases in West Africa, sub-Saharan Africa, is huge. And I wanted to help. I wanted to be at the forefront in finding cures, finding solutions to this problem. And so that was my driving force in pursuing science. And then also my faith is also a driving force, wherein with that same love, I want to have compassion towards people, and that drives some of my community involvement. So after I took my WAS, I was fortunate to get the United World College Scholarship. And these are a network of international schools that use education as a force to unite people and nations and cultures in order to have sustainable future and sustainable peace. Because when you bring people from different background. They study together, so you get to know people from all over the world. You get to have friends from different areas and different cultures that are different from you. So this is UWC, and for UWC, you do the International Baccalaureate program, and then while there, I was able to start doing extended essays. With extended essays is where you take one of your higher level courses, you find a topic that you're passionate about, and you research, write a 4,000 word essay on that topic, perform experiments. So that was another way wherein my scientific interest started building up, because back home, as some of you may be familiar with, in Sierra Leone, the infrastructure for science is not there. So this was a great opportunity. But another huge component of UWC is the emphasis on community service. So they want you to be out there. They want you to see problems in your community and find ways to just give innovative ideas to solve those problems. So I got to be involved in different community projects, both in Singapore and then around Southeast Asia, and even to Europe when we went for the Model United Nations MUN conference. So these are really some of, I would say, the background that kind of set me up for the trajectory where I am. And after you finish UWC, it gives you a very competitive advantage for college because it's a well-known, prestigious scholarship. And here I also want to take the time to encourage you, if you know anyone in SSS2 and SSS3, to share about UWC with them. They can apply for UWC, and who knows, they can be accepted, and that can also help in, I would say, setting someone up for the future. So keep this in mind. And throughout my talk, I'll be sharing resources too that have helped me, in the hope that if these are, I would say, useful to you, or you might know someone that they may be beneficial to, you can share with them. And yeah. So after UWC, I went to Colby College in Maine, United States. So at Colby, I majored in biology with a focus in neuroscience. And I was on the pre-med track. So in Dr. Andrea Tilden's lab, today I will not talk much on this, but I just wanted to set the ground rolling for that. In Dr. Tilden's lab, we studied about how melatonin protects our neurons and how that can help in neurodegenerative diseases like Parkinson's and Alzheimer's disease. And then we also compare melatonin's effect on compared to other neuroprotective elements such as glutathione, which as you may know, is an enzyme. So these are a little bit of things that I did. But then while at Colby too, under the mentorship of Dr. Tilden, I was able to do like a student genomic research course at the Jackson Lab. And the Jackson Lab is world renowned for their genomic work in providing, I would say, numerous animal models, mouse model, for the study of diseases. So these are the things that I was doing at Colby. And then unfortunately, we had the 2014 Ebola outbreak. And then Sierra Leone was one of the most hit countries. And that got me back to thinking in ways that I could help. I wanted to help, but at that time, I was mainly an undergrad. I didn't know a lot of immunology. I didn't know a lot of virology. So I couldn't help that much. And I want to pay respect to Dr. Salman Khan, which was the major virologist, also acting as an immunologist during that outbreak. He unfortunately passed away due to the Ebola. But that kind of it compelled me to start taking classes in immunology, microbiology. And so I started doing that. And I started, I would say, familiarizing myself more and more with the field of infectious diseases and how the immune system works. And so that was the, I would say, class, I would say syllabus, my major educational aspect in yeah, so the educational aspect in in Colby, but outside of the classroom or outside of the lab, I was very passionate in finding ways to impact my community, impact Sierra Leone, impact, I would say, Africa as a whole. And so as president of the Colby African Society, we organized several events like the African Soiree, where we showcase the different aspects of the continent and the diverse and rich, I would say, culture, tribe, because sometimes some people unfortunately think that Africa is a unified, I would say, body with not a lot of differences. So that is an avenue that we use to showcase that. We also did a lot of relief funds where we raise funds for Ebola, we raise funds for other relief projects. And because going to Ebola, because Sierra Leone was mainly hit, that through that I was compelled, I was passionate about giving back. And so I wrote this project, the Davis Project for Peace, and wherein I was awarded funding to help renovate an orphanage home where I grew up. And throughout Colby too, I always found ways to always be going back home to see how I can do something. So I became a founding member of PharmaCare Initiative, which is a group of doctors and nurses and other healthcare professionals that are finding ways to impact community and bringing the hospital into the community. Because back home, we know that when people mainly go to the hospital when they are really, really sick. And at some point that is too late. So what if there is not that bridge wherein people have to wait till they are so sick? What if you can just talk to a nurse? That could help in preventing the burden of diseases and even mortality in sub-Saharan Africa. So these were things that I was thinking and everything. And so after Colby, after college, I went to Harvard Medical School. And there I was working on the immunological genome project. So this is another resource that I'll be talking more about in the context. And then what I'm doing now will be mainly the research. So at Harvard Medical School, working on the Imgen, that's the shortened name for it. So Imgen is a group of collaborative immunologists and computational biologists that are looking to map out the gene expression and regulatory system. And this is a public source. This is a public source, and I'm going to also do a demonstration to see so that you can know how you can use Imgen in your context of research that could also be helpful for you. And so here at Harvard, I was in the lab co-mentored by Doctors Christophe Benoit and Diane Matisse. And Imgen, what we do, as I mentioned, is that we use different sequencing platforms to sort different immune cell types and then look at how these genes are expressed in different ways and in different conditions. So one of the ways is through the RNA-seq, which is our most famous browser, where you look at if you have a particular gene or gene that you're interested in, you can put that gene and see how the level of expression it is in different types of cells. So for that, I want to talk about one of the research that I was involved in, which is looking at how male and female, the expression of certain genes in immune cells differ between males and females. And so with this, we know that with several immune diseases, we have different prevalence in male and females. And I'll talk more about this later on. But for here, what we were trying to do is using our famous, I would say, 11 cell types. These are immune cell types, both in unstimulated and interferon stimulated conditions. We were trying to see how males and females differ. And here, most of the difference that we saw is that in females, we see a lot more higher expression of genes that are involved in interferon response. So interferon is, I would say, the prototypical, the very common antiviral response, like when you're infected with viruses. So, and then you also have a lot of complement genes, especially C3, and then you have also IL-6 and JAK-STAT, which help females mount up this greater immune response. I'll talk more about this later on. But for this, again, this is another resource that I really want to emphasize, because I know that in different contexts, or the context that you may find yourself, you may not have the resources to do RNA-seq or ATAC-seq, where you look at how the chromatins are rearranged and regulated to cause differential immune gene expression. But we have this resource and you can use it in the context of your research. So I'm going to do a demonstration on how to use this and how this might be very important. And so for now, if someone can kindly, I would say, share in the chat any of their favorite gene, or in the context of your research, what gene are you interested in? And we can see how we can use Imgen for that. Does anyone have, I would say, a famous gene? Okay, I see someone in the chat. Yes, okay. So we have, okay, well, we have, okay, we're going to go with the first one. So here, let's say you're interested in how BRCA is expressed in different immune cell types. Maybe you're curious in your research, but you can't perform it. So you can go to Imgen, and we have, like I said, any different sequencing ways, any different sequencing platform that you're interested in, you can use this to do that. So if you click on the RNA-seq, so this is the gene skyline. So here we have different cell types, and these have been sorted by famous immunologists, computational biologists, people that study these cell types. So for instance, I'm going to put the gene here. So let's say BRCA1, and I want to see how the level is in, let's say, B cells. I want to see how the level of expression is in macrophages and dendritic cell. So you choose your cell type and then you submit. So now you have the normalized expression of this gene in different cell types. And when you if you put the cursor over the cell type, it will tell you exactly what it is. So here it's telling us that it is a splenic B cell. It was sorted using flow cytometry, sorted from a 6-week-old mice, and it tells you the marker. So we see that the level of this gene is higher here in germinal centers compared to just follicular B cell. And you can see that it's not that expressed a lot in macrophages that are in the adipose tissue. So it tells you the different cell types and also the different tissue. So this is one way you can use Imgen. So let's say our famous oncogene, let's see. Let's see. 53, good. And then, okay, this is a good way, because sometimes you know that gene symbols, depending on which genome they are annotated in, they can have different variations. So you can include aliases. So if, for instance, maybe 15 years ago, a particular gene was called this way, and then it was recently changed, sometimes you can have that, wherein if you include the aliases, you're able to get what you're looking for, even though the name may have changed. Let's see. Okay, here it's saying it's not. So, for instance, the P53, initially it was TRP53. So you can see the different aliases and you can click on the one that you want. And similarly, let's look at other different cell types. So here you can see that it is highly expressed in the thymus, like the pre-T cells, the double negative T cells. And if you click on that, it can also show you how these cells were sorted using flow cytometry. So this cell type was contributed by my lab, CBDM lab at Harvard Medical School. So it will show you exactly. So this is for gene expression, but if you go back to this homepage, you can look at single cell profiling. That was bulk RNA-seq that I was showing you, but you can look at individual cell types, how that is different. You can look at microarray. You can look at different chromatin, because sometimes gene expression is also influenced by how the chromatin is rearranged, whether it's accessible by transcription factors, whether it's not. So all of these you can use here as a major tool for your own research, which I highly recommend. Okay, so going back to this, and if you have any question, again, feel free to stop me and ask the question. Okay. Wait. You see that there is this right here. I'm not sure how to get this.
Hi, Kumba.
Hi.
Oh, so you want to take off the annotation, right?
Yes, this one.
So you can just go to the annotation tool on the Zoom, then you just undo. You see a command there, so you can just take it away.
Okay.
Did you see any tool for annotation on your screen? Yeah, exactly. So.
Okay. I can't see it.
Erase pen. Okay. I think
the erase pen is not.
Oh, so sorry about that.
Oh, it's okay. It's not showing how to erase. Okay.
Yeah.
So it's only erasing for me, and I think it was from someone else, so it can only erase from that person's end. But we can continue.
Yeah, sure, absolutely.
So yeah, so after Harvard, while I was there, I mainly looked at the immune system in purely baseline state. So no disease context. But I really wanted to understand, in the context of disease, how does the immune system change? So that got me interested in the Johns Hopkins Bloomberg School of Public Health, which is world-renowned for their work in public health and also the focus of infectious diseases, which is key and very dear to my heart, as I mentioned earlier on. And here at Hopkins, I am in the lab of Dr. Sabra Klein. And in this lab, we look at sex differences in viral pathogenesis. We work with SARS-CoV-2, we work with influenza viruses, hantaviruses, and Zika viruses. We see how male and females differ in their response to these viruses. And then we also look at how age affects vaccine efficacy. So we work a lot with vaccines too. We study vaccines, and then we look at, in the context of pregnancy, how does the immune system change? And here for my PhD thesis, I am looking to dissect sex steroidal and sex chromosomal effects on vaccine-induced influenza efficacy. So I alluded to this while talking about my previous work at Harvard, but now I'm going to really talk about it in detail with my thesis. So at the extreme here, we see that male and female differences do occur in immune diseases, in a lot of diseases. So here on the left, we see a lot of female bias. So female bias, where we see a lot more females are having like rheumatoid arthritis or type 1 diabetes. And then on the right, we have a male bias where we see a lot of cancers, malignant melanomas, leukemia. We see a lot of prevalence in males. So these are extreme, but in the middle, we have infectious diseases wherein we see that male and female respond differently. In HIV, we see that the level of female is higher compared to Ebola, where male, and then for influenza, we also have females. And this is also, the previous one is for my PI, and this is also a review. She has been studying this biological sex differences for decades. And when COVID came, when eventually they started disaggregating the data by male and female throughout different countries, here 38 countries or regions, when you disaggregate the data, you see that a lot of males in blue were dying from COVID-19 than females in like the light pink. And this is the case fatality ratio. And this was calculated as the total number of deaths divided by the total number of cases for each sex multiplied by 100. And the male CFR is higher than the female CFR in 37 out of the 38 regions that they assess. And the average for male was 1.7 times greater than that of female. So this difference that we see between males and females in their response to infectious diseases, viral diseases, you can see it as both, it can be based on the sex chromosome and it can be based on hormone. So here, biological sex, so this is defined by the sex chromosome complement, so whether you are XX female or XY female. And the development of testes in males is determined by this SRY gene, and subsequently the testes then go on to produce differential concentration of androgen, so testosterone or dihydrogen testosterone. And then in females, you have ovaries that produce estrogen or progesterone, and several immune cells, including macrophages, dendritic cells, T cells, B cells, have receptors for these hormones. And these can cause different effects. So they can regulate gene expression transcriptionally. They can alter signal transduction, the downstream effect, and then ultimately the outcome of disease. Where in female, we see that females can clear the virus faster. They can mediate the pathology of the disease a lot more. Whereas in male, you have reduced vaccine-induced immunity, and they are less able to clear the virus. So that can lead to viral persistence. Another factor that influences this difference between male and female is, as I mentioned, based on this chromosome. So this is a diagram of the X chromosome, and on the X chromosome, you have several immune-related genes that are encoded. For instance, you have TLR7, TLR8, which are very, I would say, crucial for recognizing single-stranded RNA viruses. And you have FOXP3, GATA1, which are transcriptional factors, but then you also have the encoding of several cytokines like IL-13, IL-2, and IL-3. So with this X chromosome genes that are encoded, because females have 2 copies of the X chromosome, you can have X chromosome silencing, wherein one of the X chromosome is randomly silenced so that the dosage between males and females, males XY, will be similar. So female will not have 2 copies, but 1. But later on, I'll tell you that some of these genes escape that inactivation process, thereby giving females like a double dosage. So if you have a double dose of TLR7, that means you can recognize virus faster, like when you're infected with influenza or SARS-CoV-2, you can recognize it faster, and the downstream effects can be a lot more robust in females compared to males. So hence, females are better able to clear this virus than males. And previous data from our lab, here I'm showing with C57 Black 6, male and female, when they were vaccinated. As I mentioned, we work with vaccine, with whole inactivated 2009 H1N1, the influenza or flu vaccine. Females here, shown in purple, had higher level of IgG, antibody titers, than males. And similar when you look at the class switch, more effective IgG2C, female also have higher titers of this antibody than males. So IgG2C is the correlate of protection for influenza vaccine, the flu vaccine. So when you have certain titer of this IgG2C, when you're infected with the flu influenza virus, you're better able to like clear the virus and recover faster. So female produce a lot more of this antibody than males. And when we look at a neutralizing titer, so yes, you can produce antibodies, but how effective are these antibodies in actually binding to the virus and neutralizing it? So this is what we calculate with a neutralizing antibody titer. We also see that females produce a higher number, a higher titer of these neutralizing antibodies than males. So yes, they produce higher levels of IgG antibodies than males, and these antibodies are more efficient, more effective in neutralizing the virus than the ones that the males produce. And when we look at the level of TLR7, the gene that I explained about earlier that is important in the innate system for recognizing the virus, we see that in splenic B cells, so B cells from females, they have higher expression of this gene, TLR7, compared to males in green. And when we looked in TLR7 knockout animals, both males and females, we see that the level of this IgG2C, which I explained earlier as the correlate of protection, we see that both males and females, when you knock out this gene, the ability for them to produce antibody is severely diminished. So you can see it here. And also the ability for the limited amount of antibodies that they produce for it to neutralize the virus is also severely impaired. So this is showing that TLR7 or genes that are encoded on the X chromosome can be very important in how males and females respond to influenza virus. And similarly, as I mentioned, the difference can also be explained by sex steroid, like sex hormone. whether estrogen in females or testosterone in males. So when our lab also look at the levels of estradiol, which is the major form of estrogen in the body, this is in serum, we see that obviously this decrease with age, wherein in both adult females and adult male, the level of estradiol decreases and the level of testosterone decreases. And on the right here, what I'm showing is that this level of estradiol in females positively correlated with the neutralizing antibody titer. That is how functional these antibodies are. And this is negatively correlated with the level of testosterone. So then going back to the point that estrogen, the predominantly female hormone, is immunostimulatory. It can boost the immune system in responding to virus, whereas testosterone, the predominant male hormone, is immunosuppressive, wherein it can decrease the immune response. So that was the basis for my thesis, wherein for my thesis, particularly I'm asking this question: what mediates this female biased immunity and protection to influenza vaccine? Why is it that females are better able to mount up a robust immune system, that immune response, than males? Yes, our lab have previously shown that it is X chromosome encoded. Part of it is also because of the hormones that female produce. But what is the relative contribution between the two? Is it mainly because of the XX chromosome, or is it mainly because of estrogen production? Or how are these two interacting? Is there a possibility? So that is not known in the field, the relative contribution of the two or the potential interaction between the two. So that is what I'm looking at for my thesis. And to understand this mouse model called the 4-core genotype mouse model is what I've been using. So the 4-core genotype FCG mouse model has been developed to really understand and investigate the impact of sex chromosome and gonadal types, so testes or ovaries, on the phenotype. So usually on this SRY gene that I talked about earlier on, that is usually Y chromosome encoded, that codes for testes in males, it's no longer on the Y chromosome. Instead, now it is a transgene. It has been moved to chromosome 3, which is an autosome. And when this mouse is bred with a wild type female mouse, we have 4 different genotypes that are produced. So you can have the regular XX, wherein you have ovaries, but then you can also have XX. If the SRY gene is present, you can have testes instead of ovaries. And similarly, you can have XY male. If the SRY is absent, you can have ovaries. And if it's present, you can have testes. And going to this two-way table here, if we see a difference in the vertical column, XX and XY, that means primarily the female bias that we are seeing in immune response is based on the sex chromosome. But if we see it on the horizontal level, ovaries or testes, that means the female immunity, female are better able to mount up a greater immune response to viruses because of hormone, a lot more because of the hormone compared to the sex chromosome. So what I've been doing for my thesis is I have been vaccinating these 4 core genotype mice with a whole inactivated 2009 H1N1 influenza vaccine. And then I boost them on day 21. And then on day 28 and 35, I bleed the animals to collect serum for our in vitro ELISA, which is used to quantify the levels of IgG, the antibodies that I discussed earlier, and then also perform a microneutralization to see how functional these antibodies are. And then on day 42, I've been challenging with influenza virus that is a drift variant, which is slightly different from the vaccine virus. So because we know that with viruses, they mutate, we have seen that with COVID. So the strain that was used in your vaccines might not necessarily be the strain of the virus that you experience, that you're exposed to, like in the community. So to mimic that, like natural exposure, so that's why we challenge the animals with a different variant. And several days post-challenge, I assess protection via morbidity data, where I measure their weights and temperature to see how the disease is affecting them. And then also I quantify the number of virus in the lungs to see which of these animals, whether the gonadal males or the gonadal female, which ones are able to clear the virus faster. Because when you're infected with the virus, it's very important to know like how fast are you going to clear the virus, which can then influence how sick or less sick you become. So for my preliminary data so far, what I'm seeing is that gonadal females here in purple, regardless of whether they were XX or XY, they produce the same levels of IgG compared to gonadal males here in green. So, and for our neutralizing, like day 35 total IgG, not neutralizing, we see that this trend holds true, just that now the significance becomes higher. So females, regardless of whether they were XX or XY, produce more of IgG than males. And when we look at how functional these antibodies are, how they are effective in neutralizing the virus or not, we also see that females in purple produce more antibodies that are very effective in neutralizing the virus than males. So really the hormone, like gonadal sex, so like the hormone production over your chromosomal complement, whether you're XX or XY, is what is determining how effective your immune response is. And here I am showing that the pathogenesis data, so the percent body mass change. Obviously, when you get sick, I would say you lose some weight. And so that was what I'm measuring here to see how really the animals are getting sick and how it differs between these different genotypes. So for the purple, again, gonadal females, so within the same gonadal sex, so the XXF female, they lost the least percent change in body mass compared to all the other groups, to all the other three. And within the same gonadal sex, so males in green, they lost similar percent change in body mass, whereas XXF in purple and XYF, you have differences. So the impact of sex chromosome complements on how vaccine-induced immunity is generated or mounted in the body, this data is telling us that it affects mainly females compared to males. But previously, I showed you that the levels of antibodies that these females, regardless of their complements, the levels of antibodies are the same that they produce. Even the capacity of these antibodies to neutralize the virus are the same. However, we are seeing that the disease pathogenesis is different because XY females are getting sicker than the XX females. So this could tell us that on one end, that it's not one way or another. It's not only hormone-based, because if it were just hormone-based, then these two would be experiencing the same disease outcome, but we see that this is not the case. And because with XXM, the light green, and XXF, the deep purple, because also we see a difference in their outcome, disease outcome, that is telling us that it's not just based on your chromosome. Because if it were just based on your chromosome, both XXM and XXF, because they have 2 copies of the X chromosome, they should have had the similar percentage body mass change. But here we are seeing that it is different between them. So again, we are not going to just, this data is telling us that females are able to mount a greater response, not only because of estrogen that they produce, or not only because they have 2 X chromosomes. And when you look at how effective these different animals are in clearing the virus in their lungs, so I followed them for 14 days. We see that earlier on, there is no statistically significant difference between all 4 genotypes, but you can begin to see that females, the XXF females, are beginning to clear the virus faster in their lungs compared to the males. And at 14 days, so the earlier response and the later response, we see that there is no difference. But when you look at day 5, males have more replicating virus in their lungs. So males are slower in clearing the virus in their lungs in green compared to the females, where we see a significantly less replicating virus in the lungs. So this is my preliminary data for my thesis so far, wherein among FCG mice, animals that have ovaries, animals that produce estrogen, regardless of their sex chromosome complement, have greater vaccine-induced antibody titers than animals with testes. And sex hormone, rather than sex chromosome complement, predict vaccine-induced immunity in adult mice. And sex hormone might be masking potential sex chromosomal effect on vaccine-induced immunity and protection in adult mice. So for that, going forward, I have been really asking the question, what is mediating this difference between XYF and XXF? In this, I'll go on now to discuss like 2 aims for my thesis. So the research aim 1, I'm looking to determine the relative contribution of sex hormone to sex differences in influenza vaccine-induced immunity. So here is it like, once I measure the levels of estrogen in all these genotypes or the level of testosterone, could it be that XXF are producing more estrogen, which is immunostimulatory? So that is why they are better able to have a better clinical outcome compared to all the others? Or is it that these XYM have higher levels of testosterone, and that might be what is impairing their ability to have a good clinical outcome when infected with influenza virus? So that is my research aim 1. And for my research aim 2, I'm looking to determine the relative contribution of sex chromosome to sex differences in influenza vaccine-induced immunity. So really going back to what I discussed, like the genes, the immune genes that are encoded on the X chromosome, could that be what is mediating better outcomes for females than males? And with, okay, I have a question. Is it possible to incorporate these hormones in vaccine production for better immune response, especially in males? Great question, Ayobami, if I'm pronouncing it right. So I'm coming to that like in the next few slides. So that is the purpose. So how can we improve male antibody responses? So that is, yes, great question. Hang on, the next 2 slides. So here, what I'm trying to do is I am trying to see the rationale for, I would say, removing the ovaries or removing the testes so that we can clear the amount of hormones in these animals and see really the genetic components like XX and XY. So the rationale for that came from this study wherein they were using it for to study EAE, so autoimmune encephalitis. And when they used the same mouse model with gonadectomy. They saw that XX female and with here, okay, brief, let's see, okay, no, just a second, okay. So here, when they gonadectomize the mice, so they found that XXF and XYF, and then they stimulated ex vivo the draining lymph nodes with autoantigen to stimulate EAE pathogenesis and assess for cytokine production. They found that Th2 responses, especially the Th2 cytokine, IL-13, were significantly higher, like you can see here on the right, in this red box, we are significantly higher in XY than in XX females. So they decided to check the level of IL-13 receptors in these animals, because remember that one of the slides that I showed you earlier, here, IL-13 is X chromosome encoded. And when they looked in different immune cell types, B cells, they looked at macrophages and other myeloid-derived cells, they found that in XX animals, the level of the receptor for IL-13 is higher in XX individuals in red compared to XY individuals. So this is the rationale. And for my thesis, I am mainly interested in TLR7. So now going to Ayobami's question. So TLR7, as I mentioned earlier, is X chromosome encoded. It is very important in the innate response to influenza virus, single-stranded RNA virus, which influenza is part of. But then it's also very important in the adaptive response, where it is very crucial for somatic hypermutation, class switching, to go from IgDs to this class switch specific IgG2C, which is a correlate of protection, wherein if you have a certain titer of that, then you're protected against influenza infection. So I'm looking to quantify the levels of that in B cells, and also if XXF females have higher levels of these in these genes in their B cell, and also looking at how the germinal centers, like the interaction to boost the immune system, how is that occurring in this 4-core genotype mouse model. And I'm also very, very interested in looking at X chromosome inactivation, which I mentioned earlier is a mechanism where you silence one X chromosome so that you compensate for the dosage between males and females. And I'm very interested in looking at the epigenetic regulations of these genes. And previously our lab has shown that when you look at the TLR7 locus, we see less DNA methylation in the promoter region in females compared to males. So if you have less DNA methylation, so that means that region of the gene is open, you can have transcription factors that can easily bind and cause gene expression. So I'm interested in TLR7 because of these reasons, but also what I didn't get to present today is the capacity to use TLR7 as an adjuvant in vaccines to improve the male antibody responses. TLR7 has been known to escape the X chromosome inactivation process, thereby giving females 2 copies. So obviously, if you have 2 copies of that, you can recognize virus faster, you can produce the quality antibodies much more than males. So if we give TLR7 adjuvant in our vaccines, that could signifies or be as an example of giving males an extra dosage of this so that can improve their antibody responses similar to females. And so far, my preliminary data for that, TLR7 has been used in a lot of our vaccines and a lot of disease contexts, not only viruses, but my preliminary data also has shown that when we give males TLR7, like adjuvants in the vaccine, male antibody responses, male can produce similar levels to females. And this can also increase their capability not only to produce these antibodies, but the functionality of these antibodies is increased. So they are better able to neutralize the antibodies, the virus, compared to females. They are better able to have a better clinical outcome like pathogenesis when infected with virus. So this is the aim. Once we understand how TLR7 is mediating this female biased immunity that we see, then we can find ways that we can target it to improve vaccine responses for males. And this could be adding it as an adjuvant, as I mentioned. That is my other project that I'm looking at. And also this could be directly conjugating TLR7 to the virus, like influenza virus, or to other RNA viruses as well. Okay. And so for that, I want to thank, like, a lot of this work has been through several amazing people. So this is my PI, her name is Dr. Sabra L. Klein. And then also really want to thank Santosh, who is like a postdoc in the lab and has really been helpful in, I would say, getting this project started once I started in the lab. And then also I have a junior PhD student, Ina, that I'm mentoring, that is also helping me with this work. And another resource or another thing that I wanted to share is this Beyond Sciences initiative, which was a way that I actually got to meet Martin. So with BSI for short, what we are trying to do, it's an online conference that provides scholars worldwide an opportunity to present their research and network with the broader scientific audience. I remember when I was in Sierra Leone, I didn't have access to a lot of international renowned conferences. And with conferences, you have the ability to not only network, but you can also showcase your research, and then you can get feedback, and that can really improve you as a scientist. You can know what is out there in the field, you can know the loopholes and areas that you can come in with your different skills and all of that. So we organize this conference each year. So regardless of where you are in the world, you can present your work, you can meet renowned scientists from Harvard, Yale, Johns Hopkins, and people from Kenya presenting, Makerere University, from Ghana, from Peru, all over the world. You can meet people that are doing amazing work and share your work also and network. So if you can, I encourage you to register for this. And I know the deadline for abstract presentation was yesterday, but you can still submit your abstract for either a poster or presentation or a live talk, and you can email us and mention that you only found out about it today, so we can give you like a leeway. So I really encourage you. There is no harm in trying. It is amazing, and you get to learn and share your work. And so this is what I have. So thank you again for listening. And I'm happy to take any questions. And the questions can be from anything. Yes.
Wow, thank you so much, Kumba, for your wonderful presentation. I did have a very wonderful time listening to you. I think your research is beautiful, and you're doing great work in your lab. So right now we'll be opening the floor for questions for the speaker. So for those who will not be able to speak, probably they are where they are is kind of noisy. They should just drop their questions in the chat box. But for those that can speak, they should just kindly raise their hands and unmute their mic and ask their questions. So we're opening the floor now for questions now, so we can start taking questions. Oh,
I
I think
see Idris. Yeah.
yeah, Martin is saying thank you very much, Kumba. Great presentation. Okay. I wanted to ask Ayobami, apologies. Did you get were you satisfied with I think he dropped a message in the chat box during the course of the presentation. So I'm speaking to Ayobami now. I don't know. You still want to have it? You still want to ask the guest speaker any question?
Yeah. So first of all, I'd like to say a big thank you to Kumba for the amazing presentation. And I think you did justice to that question, because listening to our talk, I saw where was headed, and the fact that it is going to be a good thing to incorporate the advantage of the female hormone into vaccine production. So you've spoken basically on majorly on the RNA viruses. So I was hoping if that same method could be applied to DNA virus. Well,
Yeah. Thank you very much. That was a wonderful question. So yes, our lab focused a lot on RNA viruses, but we see similar trends in DNA viruses, and then also in parasitic infections, so like in malaria too. For the adjuvant that I was talking about, the TLR7 adjuvant that was added, we have also done, I would say, a proof of principle in malaria, because since Hopkins has a huge focus on public health, infectious diseases. So with the malaria, when it was incorporated there, we also saw that that was sufficient in improving male responses when infected with malaria. So this is a common trend, both in RNA viruses, DNA, and also in parasitic infections too. And I think, does that
thank you. So, yes. So the first time I heard about, thank you, it answers what the first time I heard about the TLR being used as adjuvant in the treatment of cancer. So I'd like to shed more light on that as adjuvant.
Right. Yeah. TLR, and then sometimes that is a great point too, in its use in cancer and other diseases. I think that has been, I would say, one of the key adjuvants that people have been looking in different contexts, and sometimes they've been conjugating it. Sometimes it's only TLR7, sometimes it's TLR7, TLR8, like a combination of it. So I'm also very curious in seeing how, I would say, that would keep increasing, like the use in different contexts too.
Thank you very much.
Yes. I saw.
All right.
Go ahead.
All right. Thank you so much for answering that question. If we take a look at the chat box now, you see that everybody, I think Leonard Francis is saying insightful presentation. Okay, I think we have someone. Idris, do you have a question? You can go ahead.
Thank you very much for the presentation. I want to actually get an idea. The FCP model you mentioned about, I didn't get it, please. Sorry for joining us, but I just wanted the recap of that model, how it works, that FCP model.
Yeah, so definitely. So the FCG, the 4-core genotype, how it works is that, SRY, which is the testis determining factor. So it's an Y chromosome encoded gene. So that gives male the ability to produce testes and then subsequently produce androgens like testosterone. So once you have XY, you're obviously going to produce testosterone. But with the FCG, how it works is that that SRY is no longer on the Y chromosome. It's on autosomal chromosome 3. So then, in normal biological context, your genotype XY is linked to the hormone that you produce, so testosterone. Now with FCG, what it enables us to do is that your genotype is not linked to the hormone that you produce. So because it's no longer on the Y chromosome, you can have XX and still produce testosterone. And so here you're able to see the effects that you're seeing in which you have a female biased immunity to vaccine, whether it is chromosome based primarily or whether it's hormone based. So you're able to see how those two interact or which one of the two is having the primary effect to the protection that you see in female. Does that answer the question?
Thank you. It does. Yes. Thank you.
Yeah, you're welcome.
Okay. Thank you, Idris, for that question. Okay. Okay. I think. Okay. Blessing is saying in the chat box that great presentation, Kumba. I'm optimistic that your work will help better vaccine production. Oh yes. So. Okay, so do we have any more questions? Floor is still open for questions. There's anyone in the chat box? And just drop your question in the chat box, or you just kindly ask your question now.
So while we are waiting for questions, I just wanted to emphasize the 3 resources that I talked about. One is the United World College, the UWC. So if you know any SSS2 or SSS3 students, please have them look into it. It's really set me up, I would say, for career success and eventually being at Harvard and now Johns Hopkins. So please, if you know, have family members or someone in your community or your church or mosque, please share that with them. And then I also shared about Imgen, the RNA-seq data browser. So that is another one. And then finally, looking out for the Beyond Sciences Initiative, the conference. So please remember those things. They have been very instrumental, and I wanted to share that so that other people can also benefit from it.
Yes, thank you so much, Kumba, for sharing that link, because I currently work with students too, so I know it will be very useful for them. So I'm going to share that with my students. Definitely, I'm going to share that. So I must say thank you for sharing that resource, because you I love great minds. I recently started working with students in my field, and I see that there are a lot of great minds in Africa, and that link you shared, I think is going to be really helpful, because I've never heard of it before. So immediately you shared it, I put it down. And for the link you shared, the Imgen, I hope I'm pronouncing that right. I think that is very, very nice, and it's user friendly too. Thank you for putting us through that. But I want to ask, I hope I can just link, I can just type it on my laptop and get it. I don't need to pay anything, or
I see Idris has his hand raised.
Oh, okay. Idris, you can go ahead with your question.
I wanted to ask, can one attend the Beyond Science initiative without having a paper to present?
Yes, definitely. Yes. You can just register to attend and meet people, hear amazing research from the US, Ghana, Nigeria, Peru. Yeah, you can attend this year just as an attendee, and next year you can think about how you can also want to present your own work so that you can get feedback, and yeah, definitely.
Thank you.
Okay, so there's a question in the chat box, so I'm just going to read that out now, and it's from Wealth. He's saying, not sure about this, but I am curious about how effective the TLR adjuvant would be in vaccine development for males. And is it possible to have cross reactions with the male sex hormone, as the higher immune response recorded in females have both genetic and hormonal links?
That is an excellent question, Wealth. Thank you for asking that. So with the TLR7 adjuvant, it's already been used and studied extensively in different, I would say, disease contexts, whether it's infectious diseases or whether it's non-infectious. So that is one way to put it. And in several of the research that I've seen out there, and also from us that I didn't get to present today, I'm working with 2 different TLR7 adjuvants, also to mention that. And in both cases, we see that when this adjuvant is used, male responses are increased to females. So we are able to boost that male response. And in the question about cross reaction and also the sex hormone being both genetic and hormonal, yes, that is, I would say that I showed you here. It's for my preliminary data that we are seeing a lot more hormonal effect. But I am curious as I move on with my thesis, when I gonna dectomize these animals, remove the gonads and seeing, I don't have an answer for that yet, but I'm also very curious to see how the genetic components, I'm beginning to see how effective or how much they compare to the hormonal effect. So I don't have like a specific answer of which one is more beneficial over the other one yet. So maybe in a year's time or 2 years' time, I'll get a major understanding of that as I proceed with my thesis. But for now, what we are seeing is that the TLR7 is very effective in boosting that, and you have very like mild or no side effects when used as an adjuvant. Okay.
Thank you, Kumba. I hope Wealth, I don't know if you you're satisfied with that.
Okay.
Oh, okay, he said, yeah, okay. I think there's another question in the chat box. Let me just go with Raji's question. He said, can you drop the links in the chat here, please? Raji, I'm not clear with that. Is it links to what exactly? Can you just clarify? I think it. Hello, Raji.
Hello?
Yes,
Hello?
yes, we can hear you. You can go ahead.
Yeah, the links to the Imgen and all those resources she shared the other time. I missed that information. I was not. Yeah, I was not there, so I wanted those links. Again,
okay.
please.
So that is the one for the Imgen, and then
and all the resources too. Thank you.
yeah. Let me just do it now. And then the Beyond Sciences, this is the link for it. And then the United World College. So this is the link for it. So these are the 3. Can everyone see in the chat?
Yes, we can see. Thank you so much. Thank you so much. So I think everybody would be able to get that from the chat box now. Thank you for sharing that in the chat box. So Abdulhafiz also asked another question. So he's saying, great presentation. I'm just curious about the potential incorporation of TLR7 as adjuvant or ligands to RNA/DNA particle. Would it not possibly lead development of genetic driven female properties such as gynecomastia? or etc. Yeah.
Yeah. So thank you very much for that question. Amazing question, because definitely when we think about vaccines and formulating a better vaccine, we have to think about the side effects. We have to think about any, I would say, unspecific target effects and all of that. So that is great. And with regards to your question, TLR7 is already produced by both males and females. So with TLR7 adjuvant, we're just increasing the amount. And so far, we have not seen any side effect that is, I would say, harmful. If anything, what we've seen is that we have a lot more immune system activation. When we use, for one of my adjuvants, when we use like 100 micrograms, so it's really boosting the immune response to become very, very effective, to the point that it's too effective. So now we are finding ways to decrease the amount, but that is mainly in the context of, we see so much activation that we don't want to cause any inflammation. So that is the only, I would say, effect that we are seeing, and that can be easily rectified. And that is what we are doing in the context of like looking at different dosage, the one that we can activate the response, but not like overstimulate the immune system. So that is the only thing that we're seeing, but we're not seeing any other, I would say, major side effects. Does that answer the question?
Abdulafiz, does that answer your question?
Okay, great.
Oh, thanks, it does. Yeah, okay. Yeah.
Any other question? And it can also be, I would say, non-science related too, and non-research. Any question?
Okay, I want to ask okay, so since the floor is open for non-science related question, what do you do for fun in your leisure?
Oh yeah, what do I do? So I love volunteering actually. I love being in the community in whatever way, whether it's like writing grants for community projects or just mentoring and volunteering. I love kids, and so I usually volunteer to help like with my church, or also just, I would say, children's centers, and working also with homeless or disadvantaged population, like homeless population, or people that are moving to the US for the first time, seeking asylum or refugee, and love like doing that. I love reading and I love cooking. I love hosting people at my place and just share some, I would say, West African meals. And I know jollof is highly, I would say, debated, which country makes the best jollof, whether it's Sierra Leone, Nigeria, or Ghana, but Sierra Leone for sure makes the best one, not because I'm from Sierra Leone, but just because Sierra Leone is amazing. So yeah, those are some of the things that I love doing.
Wow, thank you.
There is a fight. Okay, Nigeria jollof. Okay.
Exactly. I don't want us to go there at all. Nigeria jollof.
I know you guys are a lot on the Zoom call, but you can't overpower me. Well, this fight, we'll need to come back to it.
We'll definitely need to come back. Yes. So yes, thank you so much for answering that question, Kumba. Yes, I think we'll just give a minute of you for anybody. Okay, Goziti. I think Goziti is raising her hand. Okay, Gozi, you can go ahead, please.
Okay, good evening, Kumba. My name is Chigozie. You can call me Ruth. I think that's easier. So
Okay.
I actually, from the bio, I think you obviously you've gone to Harvard, you're in John Hopkins. So since this is not science related, but I just wanted to know, those are for us big names. So what is it like for you being there? Is it as rigorous as people like make it seem? I just, it would be nice if you could like just share it, like your experiences or something that just breaks the stereotype that people have
Right.
about it.
Right. Yeah. That is an excellent, wonderful question that over the past years I've actually been introspecting. Definitely these places they are definitely big names, and because of, I would say, research funding, and because of the environment, they can be intense, in the sense that, yes, you need to, I would say, work hard, you need to put yourself out there. But one of the major things that I personally got from it is that at the end of the day, yes, your intelligence will take you far. Yes, you have to have good grades. Yes, you have to score well. Like I mentioned, I was the head girl back in Sierra Leone for my school. Yes, intellect take you very far. But once you get into these doors, at the end of the day, your intellect stop at that point. Now what really matters is your work ethic, how you treat people. Because what I mean your work ethic is that you can be smart, but if you're not hardworking, we all know that research or pursuing a higher degree takes perseverance. Like things will not work. Experiments might not work the first time. So are you going to persevere through that? Are you going to keep showing up? Are you going to be diligent? So those things help. And then secondly too, how you interact with people are things that keep you in these spheres. For instance, again, I got to know Martin through BSI, and BSI got involved in BSI because of someone that we shared this similar lab bench. And once you have good relationship with people, you can share ideas like quickly. When things are not working, you can discuss. And I am certain that when you discuss, when you share your work, you get a lot of feedback. You may be looking at one particular way when other people, just because they are seeing it for the first time, they have a fresh, very fresh perspective, so they can give you that. So I've seen in these places that when you're not a kind person, that can hinder the amount of collaboration that you have. So yes, it is. Yes, it is intense in that sense, but it's also like very collaborative in certain areas, depending on your interpersonal skills with different labs and different PIs. And another thing with these institutions is the amount of funding that they have. There could be an exceptionally brilliant person, and there are brilliant people in Sierra Leone. There are exceptionally brilliant people in Nigeria, Ghana, Burkina Faso, that are a lot more intelligent than a lot of us here. But the difference is the resources. These institutions have been in existence for a long time, so they have the money for fancy techniques like RNA-seq, like some of the experiments that I used to do for just one plate, it's like $10,000, and we didn't even blink. We can do like multiple, we used to do multiples of those a month. So in as much as they are great, in as much as they are wonderful and all, I think the funding is another part of it that makes them exceptional, but that doesn't mean that other institutions are not exceptional, just that the funding is not there. Does that answer the question, or is there another particular aspect you want me to answer in that light?
Yeah, it answers the question. Okay, I'm sorry, I'm going to ask another one. Now, aside from the environment, the requirement, the way the institution is, what was it like for you personally coming from
Oh,
first
from
Africa?
theory. Oh,
Yeah,
yeah.
and then just going, and just going into that environment, I think I also want to know how you felt and how you've grown up. Maybe tips that you could share with us concerning like navigating that place.
Right. Okay. So the in light of that, the funny thing is that what most people don't know, and what I also didn't know, is that the Harvard University, the undergraduates in the Harvard Medical School are not in the same location. So for my interview, when I went to the medical school. I was supposed to go to the medical school. That is where my interview was. But then just because I didn't know, I went to the undergrad. And then I realized that they are in different locations. But I came like 4 hours early. So I had more than enough time to make it there. And they gave me the visitor's badge and all, and I looked at it, and then I looked at my name on it, and then I was like, wow, I'm a Christian. So I'm like, thank God, I've made it to Harvard. So I'm like, even if I don't get accepted, I've made it. My name has appeared like on a Harvard badge. So there was that feeling of like, I would say, wow, God has brought me this far, like from Sierra Leone, and then now I'm here in working with people that are, I would say, dictating what is taught in medical school. Because my boss is, he discovered CTLA4 in the context of the immune system, and we know that CTLA4 is huge. So I couldn't imagine that there was this other one time, if you stay at particular time in the evening, my friend worked with someone that actually won a Nobel Prize. So there are times wherein it's surreal. You're like, wow, I'm interacting with these people, and I'm from Sierra Leone, and Sierra Leone is like very underdeveloped. So there is that part of it, but there is also the part where I feel an extreme sense of responsibility in finding ways in whatever manner to always give back to my country, to always give back to Sub-Saharan Africa, tailor my research in ways that could help, and also share resources, things that have helped me. Maybe it could help another person. So there is that sense of responsibility. So that was why I was so I would say humbled when Martin reached out. And then I told him that it didn't matter how many people are on the Zoom call. For me, even if it's one person and I'm able to share what has helped me, if that person can incorporate that, that is a win for me. And so that is the kind of responsibility that I feel. And then also the responsibility to I would say, communicate science in a way that is very effective. Because the moment you talk about these big names, or people know that you're from these big names, whatever you say carries weight. Because people, I would say, respect these institutions. And we have seen that with COVID, and especially with the vaccine, even here in the US, people in the faith community, and then people of color, especially the Black community, have distrust for science because of several things that have happened. For instance, the Tuskegee effect for syphilis, wherein the incorporated Black men in that study, but even when treatment for syphilis was out, they didn't offer the treatment option to these people that were in the study. So that was how distrust in science began. So now, as a Black person and as a woman also in science, in fields where sometimes I'm the only Black female in the room, I feel that sense of responsibility to communicate science in a way that I would say is true and also very effective, because I cannot be doing something, I cannot be doing science, and communities that I associate with are suffering. For me, science is about people. Science is about improving the lives of people. And so being at these big name schools have given me that opportunity, that platform to also share that. Sometimes when I go to school in the morning, if I take an Uber, the Uber drivers, like, I've had amazing conversations wherein the Uber drivers or Lyft drivers would be like, so are you telling me that I should take the vaccine because you say you work on vaccine? Do you trust the vaccine? So I'm like, yes, again, I was so fortunate because our lab works on SARS-CoV-2. So we were among the very first people in the whole world to get the vaccine because we work with live SARS virus. So for me, that blew my mind that of billions of people in the world, I was among the ones that were privileged, even before it became open to people here in the US. And so having all of that, all those experiences and being in that Uber, and that Uber driver who was Black was, do you trust the vaccine? So I told him that I have taken the vaccine. And I told him about the side effect that I felt. I told him about my opinion and all. It was not like big science. It was not like, oh, TLR7, innate immune system, adapt. No, it was not all those jargons. It was one person communicating to another person. And I am not sure if that Uber driver went on to take the vaccine. I am not sure. But I am certain that, yes, that conversation was meaningful. It could have also been meaningful, not only for him, but for his family too. And for me, that is science. And that is what being in these big name schools have offered me. So does that answer the question?
Yeah, it does. Thank you so much. Thank you.
Oh, thank you so much, Robert. And thank you, Kumba, for taking that question. So we are going to, in the next few minutes, we'll be rounding up. Or before, I saw Dolapo wanted to ask a question. Please, Dolapo, kindly ask your question now, please. And we have one more question in the chat box. So we're going to take Zion's question after Dolapo, and we're going to round up to this session. So Dolapo, please kindly go ahead.
All right, yeah, thank you so much, Olaito. And thank you, Kumba, for the awesome presentation. It's been amazing listening to your stories and your research. So I just want to ask a question. I mean, this is outside your research. Yeah, so I wanted to ask about the program for SS3 students. Yeah, so I'm wondering.
Please, I want a question.
Sorry, there are interference. Can you mute your mic?
Hello, can you kindly mute your mic? Please, everyone should ensure their mic.
All right, okay. Yeah, so I wanted to know if you did any tests, maybe SAT or something, at the secondary level for you to secure that funding. Yeah, so this will be enable us to communicate better to people who want to apply to that funding opportunity. Thank you.
Yeah, you're welcome. So with UWC, I didn't take SAT for applying. I only use my WAS. So my SS3, so that's the only thing that you need with your, I would say, regular school report card, and then some recommendation. So that's the only thing that you need for UWC. And you can talk about some of the community involvement that you've done. A lot of people in West African countries volunteer with churches, volunteer around communities and everything. So that is the major thing. You don't need SAT, you don't need the TOEFL. No, you just need your WAS, or sometimes you can even apply, like I said, in SS2. So even before taking your WAS.
Oh, perfect. Thank you.
Yeah, you're welcome.
All right. Okay, I'm just going to be reading out Zion's question in the chat box. Okay, yeah. Saying does sex-linked immune differences have any correlation with reverse vaccinomics?
Yeah, so that is a wonderful question. And so far, we have not seen any major correlation throughout the clinical studies that have come out in the pandemic, any link or correlation between sex-linked immune deficiency with reverse, very, I would say, dire outcomes for the vaccines. But what we have seen though is that is repeatedly brought up is that female experience more adverse, used loosely, reaction after taking the vaccines than males. So like some female will experience higher body temperature. Or for me, I slept a lot. I slept like extra 4 hours the night that I took the vaccine. And some may report like slightly longer headache, maybe like a day or two. And some of the immune, I would say, effects or correlation that that was also mentioned is not really sex linked, but it's mainly, I would say, in the context of severe comorbidities. So like cardiovascular or I would say diabetes, but not specifically sex-linked immune differences. Does that answer the question?
Zion, does that answer your question? Okay, yes. All right. Thank you so much, Kumba, for taking all these questions. Okay, just to be sure that there are no more questions or concern, I just want to give one more person to ask Esau a question. Just one more person before we round up tonight. Okay, Idris, we'll be taking Idris now. Idris, please kindly go ahead.
Thank you very much for the session. It has been amazing. I want to ask if there are rooms for mentorship as to how one can actually get into some of these big school names, and also probably a PhD or a master's degree program there.
Yeah, definitely. There are several opportunities. I have a couple of, I would say, avenues like Beyond Science initiatives. And then I also know friends that do Young Achievers Forum. But personally too, I am more than open to read any personal statement, to recommend schools in terms of the research that you're interested in terms of areas that you can do for funding. The only note is that with the US, to get a PhD funding, or with PhD, once you're accepted, that comes with funding, whereas for masters, it's harder to get funding. So that's the only consideration. But feel free to reach out to me if it's an area that I am directly linked to. I have, I would say, the skill set and resources. I am more than willing to help. But if I know a friend that could be better suited to help in another area, I can link you to people, and also to, I would say, professors too. People are more than willing to help. Like just reach out, and LinkedIn, you can reach out to me through LinkedIn or Twitter, and I can share resources. Yeah.
Thank you very, very much. I'm grateful.
Okay, thank you so much, Kumba, for taking all those questions. So on behalf of the community and on behalf of Science Cafe, I would like to say a very big thank you to you. Thank you for your time. Thank you for effectively communicating your research with us tonight. And thank you for joining us tonight. You have, I'm sure you've definitely inspired me and inspired a lot of members in the community that's joined us tonight. And you've also shown that research is beautiful. Your research is actually really beautiful, and it's really nice to see that there are these differences that you have shared with us in the immune response to viral infections and vaccinations. So we definitely wish you all the best in your future endeavors and with your research going. So thank you so much for joining us tonight, Kumba, and thank you to everyone, the members of the community, for joining us tonight, and thank you so much for your time. It's a wonderful session. Yeah.
Thank you all. I'm truly humble, and thank you for making time to listen to me. Thank you for the questions, and I really enjoyed it. And I also hope to be in the audience in future sessions. So I really appreciate it. Thank you, everyone.
We would love to welcome you in the community very soon. So thank you so much. Yeah. Good night. Good night, everyone.
Good night. Hello? Can you hear me?
Hello?
Yes.
Yes, we can hear you, Martin.
Okay, yeah, I just want to say a few words to add to what Olaitan has already said. Kumba, thank you so much, and we really appreciate your presence. And yeah, we'd love to have you again, and we'll talk further, especially with respect to the other resources you shared. I think there are some other things you've been interested in, and we'll talk more about it. Thank you so much.
Yeah, that
And
sounds
yeah,
good.
I loved your presentation.
Thank you. Thank you for listening, and thank you for having me again.
Our pleasure. So have a great day, Kumba. Good night, everyone.
Yeah, good night, everyone. Thank you for joining us.